Pharmacy practice. Okay. Okay. Good, good. Anyway, any other person is left, I think, maybe. Okay. I think it looks majority of them are, I mean, of you are all from pharmaceutics. Huh? So there is more demand from side for pharmaceutics, something like that. Huh. Okay. Anyway, um, see, um, first I was thinking of talking something about how do we go about a drug discovery program, but then I thought since everybody is faculty, I don't have to go into those things. Instead, what I'm trying to do is I will talk about the areas where we have worked in as examples and how did we play around building molecules because you need to have new molecules to develop them as drugs. So this is where I'll go one by one by giving, but before that, I'll give a little introduction about, since I'm from more medicinal chemistry aspect or the pharmaceutical chemistry, uh, what exactly, how this whole medicinal chemistry got evolved in the last several years. See, in the long back, what we call it as the early Iron Age, uh, many of these things started. How to do treatment for various problems, diseases. Because those days, diseases were practically not studied that way. It was only based on certain symptomatic conditions. Somebody has something. I mean, this is what was the understanding those days. So, and you can understand that Ayurveda, Egyptian medicines, Babylonian medicines, and these Chinese medicines, they're all from, again, natural products mostly. And they were into practice. Then 1025 AD, this is where this is called as the medieval period in the evolution of the medicinal chemistry. And that is where the Unani medicines and medieval medicines started. So these are the things, but 19th century brought a big revolution in the drugs, pharmaceuticals, and how really the medicinal chemistry has to be done. And what you can see there, again, it started mostly with natural product like molecules, then based synthesis. These are the again the natural product based molecules, and also you synthesize molecules that are very much alike to natural products, so that then you have uh, scope to do much more. Because natural product is something where you isolate a particular product, but it is a single entity. But when you are synthesizing, you can always make larger number of molecules where you can have different permutations, substitution patterns, and things like that. So this is where 19 things look. But if you now look into the 21st century, what's happening currently is we have rational drug design like HTS, nanomedicines, what we are hearing every day, advanced drug delivery systems again, and targeted therapies. Because therapeutics through nano, you can, you know very well, you can reduce the dosage like anything. And so that it becomes much more available. The olden days, you may be remembering, it was something similar to that, and painkiller which is called as with the name Aspro, which was very popular in probably our childhood days. Then they started with microfined Aspro. So it's the aspirin which is microfined in a powder form. So it is something little below my nano. So it happened long back, that also, where it was formulated in such a way that the particle size was much smaller. when they used to material. And that's how it became very popular, because the action of this microfine aspirin used to be faster than the ordinary aspirin because of the particle size difference. Because as you understand, the bio availability will be far better off, the absorption goes well. So these are the things. That's how nano is also helping us. And targeted therapies, as you know, are also very important today, because in many disease conditions, that the drugs could become very toxic on long-term usages of cancer. Where therapy now, although it compared to the normal drug dosage. So, and also with also. So these happen today. Although I'll give you some examples. How do we first design a molecule, probably potentially become a drug? So I try to touch in the area of cancer 2 or 3 target aspects. Cancer, as you know, it's a really an issue today. I mean, where a large number of population, because aging population is, although we have had a lot of, I would say, medical interventions with respect to people can really improve their health after certain surgeries and things like that. But in case of cancer, this has been a very complex issue, because it's not a one disease type. It's a combination of so many things, and it's of so many types. And this is where one has to really do the treatment, where the numbers are becoming very high today, close to almost 19.3 million and deaths worldwide. Which are used as therapies. But the 3 important ones, which are major ones, are chemotherapy, radiation therapy, and surgery. These are the main things which today people practice for treating cancer. So now, to explore chemotherapeutic agents, how do we go about? They are of different categories. They are classified based on various, how do they interact a particular chemotherapy drug with with a receptor. So, and many of these have to do something with, as you say, DNA has a central role. I'm sorry, I just take some. So with this, you can understand we have covalent binding agents. Then we also have non-covalent binding agents. Then we have various other inhibitors like trifolate, ribonucleotide, all these. And also in turn, there is something through DNA, RNA also, where through proteins, what all these type of agents like certain enzymatic and hormones which are used, and also the chromatin function inhibitories, which is again a very important one, which is getting lots of importance, that is the microtubule inhibitors. I'll show you to that once we go into the details of each aspects, which I'll be covering in this. See, these are very common natural products which are being used. I mean, most of you, I'm sure, are aware of uh, as anti-cancer drugs. Taxol is very much in the market since quite some time. Then we have Toptecan. So basically, Toptecan is nothing but it is a natural product, camptothecin, which has been slightly modified because camptothecin went up to phase 2 clinical trials. And what happened is it didn't have proper bioavailability. So as a result, they had to come back and then look into how do they modify this molecule, and that's how Toptecan came. Even irinotecan is there, another which is in the market today. Then this is again a very common plant which you find a very hardy plant in our these things. This is again a complex molecule. All these things, as you know, including doxorubicin, are treating cancer, but with a cost because of the heavy side. Molecules are natural, but highly toxic. So based on that toxicity, it is killing the, but they can accelerate cancer. So as a result, you get severe side effects. So it kills not only the cancer cells, but also kills the working on cancer cells. So lots of research has happened in the last, I would say, particularly in the last 20 decades. These are again couple of again molecules which are again natural products. I'll be talking something more on this. This is anthromycin, which is from a family of pyrrolobenzodiazepines. It's a tricyclic ring system. And these antibiotics are basically isolated from Streptomyces species. And similarly, you have other things like combrestatin. Combrestatin is basically a molecule which went up to, again, phase 2, phase 3 clinical trials, but again, issues. So people started trying to make newer structures, I mean, with newer structural patterns on this scaffold. So that is also, I'm going to speak today, something on that. And what you see here is this particular molecule, which I'm going to talk in detail, how the mechanism happens. I will talk to you once I start with that. And there is another molecule, which is etoposide. So etoposide, you know, is a semi-synthetic molecule of what? Photophyllatoxin. So photophyllatoxin is a natural product. So to make it soluble and bioavailable, this sugar aspect has been added to that at the 4-beta position. And that is what is the etoposide. And this is also in the market and has been extensively used. Then there's another simple molecule, which is a beta-carbon derivative, harmine. This is again a natural product. And a lot of, again, molecules have come up. So for instance, this is the DNA minor group binder, and this is a microtubule inhibitor and disruptor, and this is a topoisomerase 2 inhibitor, and this is also topoisomerase 2 inhibitor. Now, these are a couple of areas where I don't go into all this. I will be speaking only on this aspect, mostly pyrrolobenzodiazepines, where with photophyllatoxins, scaffolds, and comprostatins, and some other miscellaneous scaffolds, I will try to brief you as early as possible. And then once you think it is enough, I will stop it and try to pack it up. Okay. So as I mentioned to you, these are some important target aspects with respect to the development of anti-cancer drugs. And these are the couple of examples which I've already mentioned. Coming to now pyrrolobenzodiazepines. See, this particular scaffold, if you see, this is not known naturally, but it's the basic skeleton of this heterocyclic skeleton. These are all the naturally isolated molecules from various Streptomyces species. And one of them happens to be the anthromycin, tomamycin. These are the two molecules which also went up to phase 2 clinical trials, and they had problems in the third because of mostly cardiotoxicity. Then even ticamycin, cybromycin. So cybromycin was also brought in to improve, was also used since it has a better bioavailability. Now, coming into the mechanism of such molecules, these particular molecules, they basically get inside the minor groove. See, DNA has a main groove and a minor groove, the helical structure, so you all are aware of this. And these molecules get inside this minor groove, and they have a covalent linkage that you will come to know how it's happening. So this is the proposed mechanism, and in fact, a lot of studies have happened to practically prove this. Here, it also goes into the imine, this type of an imine to carbinolamine. It goes into this particular scaffold. And once it is in the imine form, the NH2 of the guanine, of the DNA, you get a covalent linkage. This is what you found, an aminal bond, and this is the compound. See, the basically the other to make a mention is this type of they span for at least 3 space. So this is where most of us started looking into how we can have that selectivity. If the binding can be a little selective, then whether it could differentiate between a cancer and a normal cell. So this is where the preference is between purine, guanine, purine, these 3 base pairs when this happens. And this is only a model where you can see this whole yellow thing is nothing but the anthromycin, one of the example of this PBD, which is docked inside the minor groove. And this is where you can see how nicely it goes and sits inside the minor groove. The space which is available really allows that. There are a lot of things which have been studied in this. I'll try to cover a few things. First, I'll run through these because synthesis of this pyrrolobenzodiazepine system is very, very critical, because you have to have an imine functionality intact in this. See, having just 3-ring scaffold is not a problem, but the imine functionality is supposed to be the highly reactive functionality. So that is where it's very critical way you have to prepare this. So the synthesis is not that simple, although this is a mistake. This is Lawson's reagent, not Lesson's. And this is a general way it is synthesized. I don't go into details of this. So this amine in the presence of methyl will become the methyl ether of this. And these are the different approaches. See, whenever you are talking of synthesis for the physical chemistry people, you know that you have to think in a retro way. What could be the starting materials that can be used? What are the? So you try to break down that molecule and see from where you can start. So these are some of the starting points. You see 1, 2, 3, 4. These could be the starting points for building that ring system. So this is another approach. In fact, this is done by one of the group in UK. In fact, we became collaborators at some time. And this was the approach developed by him and his group is a synthesis of this pyrrolobenzodiazepine with the imine functionality. Then we have, there's another Japanese group which worked on through this method. See, every method has their advantages and disadvantages. The end yield is very, very low comparatively, because to keep the imine with intact, this thing, as I said, is not easy. So these are some protection, deprotection, FMOC things are being used to get this. Then we developed a couple of approaches using these protocols, and through these type of precursors, what you see here. Then these are, I would say we must have developed something like more than 2 to 3 dozen methods to come to this scaffold, so that we have everything handy when we want to make our new molecules. Which method will work good for which, we will not be sure. So that is why first we did a lot of exercise for a couple of years to synthesize this scaffold by a variety of reagents, variety of systems and things like that, which can go better. And we have that whole listing here, and some of them I have just given here. We also did some things through the solid phase synthesis also, because those days we had the synthesizers where you put your things and then you can make a library of hundreds of compounds in small amounts, and then through the solid phase way, through the synthesizers. So that is where certain experiments were planned, and we were quite successful in making libraries of such molecules. Then this is another thing which, when we are talking of more of green chemistry, we want to recover most of the things as much as possible, particularly the reagents. So these are mostly I would say, the resin-based reagents what have been used in this particular protocol, and where these reagents can be recovered and reused. Now I'll come to the structure activity, because this is where we develop this structure activity relationship aspect, which is very important when you want to develop any new molecule, because you have to first understand that what particular aspect in that structure or a scaffold is important for providing the activity. So those aspects you should not disturb at all. You can play around with certain other aspects where the particular substituents or particular points are not disturbed. Otherwise, you lose the activity. I'll just talk about 2-3 brief things. As I mentioned to you in the beginning, we are the N10C11 position. The imine functionality or the imine ether is the utmost requirement. If you don't have this particular imine functionality, the molecule is practically not at all active. It's zero activity. The second aspect is the stereochemistry at C11A at this point. See, I must tell you, this particular hydrogen, when you have this 3-membered ring, benzene and the 7-membered ring and the 5-membered ring, what really happens is, it's the 5-membered ring goes down like something like a half-open envelope. Okay. You have an envelope, its lid will be like this, so it's like half-open. So that is the shape it will get when the hydrogen sits on the top. Okay, so that shape is very essential for it to get inside the minor group. That is why these two things are very, very important. The other things I won't get into too many details. The only other thing is, you can have certain type of things, and you can also have modifications at C8 position, where you can link with many other things without disturbing the activity. If you do it in this space, what happens is, when it goes for covalent binding with guanine, if there is a bulky thing here, it will come like an oxidation. So such things should be avoided. Okay? And these are the other things I want to go to many. Now, I'll give you an example which has come up from the Thurston's group. This is, you have seen the pyrrolobenzodiazepine. Now this is a dimer of pyrrolobenzodiazepine, which is with a spacer. It's an alkane spacer. So as I said, that C8 position is okay for attaching things. So what they did is, they designed such a molecule so that the single amine in the molecule has a 3-base sequence pair. So once you have 2 amines in the same molecule, what was expected was that it will span out the 6 base pairs. So that is where it has been mentioned that in case of these dimers, the 2 active sites, that is the 2 amine functionals, are responsible for the cross-linking capability of the to DNA. When it goes inside the DNA, with the 2, basically it's a cross-linking with the 2 strands of DNA. So that becomes much more effective then. The DNA binding will be highly effective. This is what they designed. And you can see that one of these molecules which went up to phase 3 clinical trials, you can see the type of activity it has. I mean, it is highly active, means highly cytotoxic. So, and also they had selectivity with G sequences in DNA and also low affinity to AT-rich sequences. These are some of the things. And the DNA and cross-links it, as I said, the 5' purine and GAGC pyrimidine sequence. So this is where you have improved the sequence selectivity. Now, there were problems, as I said, it went up to no doubt phase 3. But we thought of how do we, because there was a little bioavailability issues and also solubility issues in that dimer. So we tried to design a molecule by bringing in a piperazine group in the spacer so that we don't disturb the imine aspects. The spacer will become a little longer, but this particular scaffold is very well known to improve the bioavailability. So what we did is, after doing this, we can compare this with the molecule which was prepared there in the previous slide. So you can see the results, what you see here, these are the thermal denaturation studies. What maybe I should briefly tell you what is that, since it may or may not be known. How do you look into DNA binding aspect in vitro? This through some calf-thymus DNA, try to do that. And how do you do that is, it's a thermal denaturation of that. That means you use this calf-thymus DNA, you add your compound in that, what quantities and all that, there are some details in that, I don't know it. And then you bring the temperature up. So once it goes up, see what happens is, when the molecule is sitting inside the minor group, when you start this thermal denaturation aspect, you're giving the temperature up. It's a UV equipment which has all this in this. So you see all this. And something like, if you take an example of a zip, when you open the zip, the 2 strands come out. So it's something like that. When you do this thermal denaturation, it happens that way. And that is where the more the temperature it is going to, okay, the more the difference in the temperature, that is delta Tm, we call it as degree centigrade, is value. That means it is much more effective. That means the binding is very tough. So we had molecules which had far better binding than the, you can see almost double compared to the molecule which was already synthesized. And this is where we could improve upon. And this is the modeling aspects what we have seen. This also has a cross linking activity, but it is not only improving the bioavailability, but it also improves its binding ability also, which has doubled because of this piperazine ring inside this spacer. Okay. Now, we have also, see, when we were trying to understand that this cross-linking is happening, and is it the only thing which is giving this enhanced DNA binding ability, or is it something different? So then we thought of synthesizing molecules where one ring will have imine in this dimer, the other one will not have imine. So that whether with this one imine also with this, without cross-linking, how effective such molecules could be. So when we did this, that is what we call them as the mixed imine-amide dimers. And even these molecules, one of them, based on the spacer length, had almost very good DNA binding affinity. So here we try to discuss upon, this is a very detailed thing which we have done in, that it's not the cross-linking itself, it's the non-interactive other the other interactions with the DNA of the molecule, non-covalent interactions, they also play an important role. This is what we try to conclude, so that the DNA binding ability is almost close and almost better than that in this case. So this is again the modeling aspect, what you see for this when you put it inside the DNA. Now, we were thinking, although people were making in the monomers some substitution variations and the dimers, but we thought, why can't we make some sort of hybrids and conjugates of this pyrrolobenzodiazepine with other scaffolds, which also can play a role in the interaction with DNA, so that whether we can get much superior molecules. So here are a couple of examples where the pyrrolobenzodiazepine has been linked to benzimidazole type of conjugates, which are again very important DNA interactive compounds. And part of this has also come out in a drug, chemotherapeutic drug. So we try to use certain scaffolds of this type. So you can see how good the DNA binding ability is happening just with pyrrolobenzodiazepine and something substitution patterns and all that. And that time the company Ranbaxy approached us. I won't say completely, most of it would be, and a large number of these molecules were, I must mention, were studied with the National Cancer Institute for the 60 cell line assays. So then we started making newer molecules in based on the scaffold of photophyrotoxin. Now again, to make new molecules, as I told you, to having a proper molecules which could be of value. So without going into too many things, these are some important things where this trans stereochemistry is important, this beta is important, and again this type of dimethoxy is important, and free rotation of this type of a E-ring is important. But where do we find things to do were brought in? I mean, these are something like these are not very huge chemistry, but only thing is this beta position has to be intact. That is what is more important while you are doing any reactions. And also some polyaromatics. So many things were done, and what we could see is the IC50 or GI50 values or whatever it is were far superior than etoposide also in many of our aspects of photophyrotoxin, which we did, and gave some interesting results. Here also, I mean, you can see that the GI50 value is 0.2 and all that. So I can continue for another 15 minutes or so. Huh? I hope it won't be boring to you, but still, I'll give you as much as possible brief without going into too many technical aspects. And around this same thing, when we are trying to do changes at 4-beta, we wanted to bring some sort of an amide aspect through an aryl linkage or an alkyl linkage or direct another immunolinkage. These also give, you can see, very good GI50 values. Now, This so many things were done on this, and uh, here what you see is, I must tell you that when you go for such scaffolds, um, there are also possibilities that you may get DNA topoisomerase 2 as well as topoisomerase 1. So we checked both the aspects, and these molecules had a dual activity, in fact. Similarly, this is another example where functionalities like ureas and all that are in place, which because we thought it can have much better interactions when it is inside that pocket. And these are totally synthetic molecules, keeping the scaffold of. Um, so we talked about. I mean, we prepared the 4 AZA type of functionalities in this, the hydrophotophlotoxin. And as you know, you need to do a lot of studies relating to that, whether you go for analysis of tubulin polymerization, assay, and so on. So these are again a couple of examples where one has used alkyl amidochalcone, beta cinamido linked phyllophyllotoxins. And what we could see is these were quite good apoptotic agents. You know about apoptosis, most of you. So such molecules really uh, were highly apoptotic, and that is where they were all examined in detail. You can see 2 of them, which are being done through this hex training and so on and so forth. And we could, I'm not getting into this because this visibility is very low compared to, I don't know, it's the light problem or something, resolution, how they behave here. It's compared to, again, uh, the control and also the etoposide and other molecules. These are again a couple of other molecules where the modeling aspect also is taken, is gives you this thing on the DNA topoisomerase 2 alpha, which induces apoptosis also. And many of these studies, I'm sure you know that how you carry out the Western blot and for understanding the topoisomerase. Because nowadays, all these things have become much simpler because kits are available now. Good GI50 values. And these molecules also inhibit microtubulin assembly. So it is not topoisomerase, but it inhibits the microtubule. So these are certain exceptions. Once you make them structurally totally different. So these are some, you can see, I mean, these are just examples of how we have tried to modify with different these things. This one is also another modification which induces, again, apoptosis. So a lot of details have been done from the biological point of view. Uh, and these are, again, triazoles are also very well known, either 1,2,3 triazoles or 1,2,4 triazoles. In many of the cytotoxic and uh, uh, chemotherapeutics, this scaffold has also been used thoroughly. So we have tried to combine them with the phyllophyllotoxin scaffolds. So this is again the thiourea type of a functionality one would bring in this, and also see, which has also given us some promising results. This is a inhibitor, which is again tubulin inhibitor, where you can see this is in true sense not phyllophyllotoxin, because this is totally synthetic molecule what we have made. Where you don't have that, but it has some sort of an I would say it mimics the phyllophyllotoxin left hand side rings, uh, A, B, C, and E. This is again the aryl urido type of molecules were also made, where we wanted to make more of the urido and amido type of functionalities to improve interactions. Similarly, we have this amido, 4-beta amido ones with these type of heteroaromatic link type of. These are just some examples I'm running through with all interesting results. where we had some selectivity for certain colon cancer and things like that. Now, so tubulin polymerization I was mentioning is another important aspect apart from the topoisomerase 2 as a target. And this is where you can see that you have in tubulin polymerization two aspects, where you have stabilizers and destabilizers. And you have different type of these, what we generally call them as the paclitaxel or vincristine or colchicine. These are the binding sites. These are well known, and so you try to compare when you make some new molecules with these sites where they are going and fitting in the tubulin. So these are a couple of molecules which have are well known for the tubulin polymerization inhibition aspect, and this is a totally where we could improve these molecules in such a way that it has gone in for much more detailed studies in our study, I mean, in our investigations. So this is combrestatin A4. I think quite a few of you may be aware of this combrestatin A4 also. This is a very interesting natural product. And here also, we have done a lot of work, and this is where the structure is. And this is a lot of changes can be done on this for modifications, and this particular ring particularly. And we, in fact, we had come out with a very, quite recently only, I mean, 4 or 5 years back, we came out with an excellent review on the therapeutic patents of this class of molecules. Because we have reviewed many of these things in different ways. And in that, we could come out with some interesting aspects, how the scaffolds are prepared. These are molecules with all variations. And if you see, if you go back and see this particular scaffold, we have tried to keep it in such a way and also bring in other heterocycles and other things to have some sort of an replica of that particular scaffold of combrestatin, and whether we can improve its. This molecule also had issues. This also went into clinical studies, but could not proceed further because of the bioavailability issues. Because many of these natural products have bioavailability issues, and a lot of groups started working on this, and ours was also doing lots of work in this. And I must tell you, in fact, yeah. So such type of scaffolds also were designed, synthesized. What you see is there are certain amino benzothiazole type of conjugates we have made, because amino benzothiazoles also have this particular tubulin polymerization inhibitor. So we wanted to bring in scaffolds which also does that. So whether it has any advantage or not, we wanted to understand. And also we have made efforts to make them bioavailable, keeping the substitution patterns in such a way. So I will just run through this couple of examples what we have done. Very interesting results have come, and many of these very good structure activity relationship aspects were also understood, including the modeling aspect. So these are some molecules which were again designed keeping in view this particular thing. If you see, having a sulfonyl piperazine in place, and this is where some very interesting biology came out for us, with respect to its not only its activity, but also the way they interact and all that. These are some details of that. I don't go again into details. This is again fenstatin is a basic system, which is again an important one, where people use different scaffolds with different modifications. And we have done quite interestingly, uh, bringing fenstatin as well as isocumbrastatin oxindole type of a conjugate. So if you see, each component has been brought into one scaffold. And that is where we could get some very interesting results in this too, including the tubulin inhibition activity. So this is another scaffold, which will be the last one I would just very briefly say, the carbolene, beta carbolene. And this is a very simple molecule from natural product point of view, not very complex. And in fact, we did, I would say, again, a large number of scaffolds that has been built in this particular drive. And must be, I think, close to 30 to 40 new scaffolds were brought in, utilizing this carbolene structure. And to my surprise, somebody came as a review, which was dedicated in my name from a professor from Punjab University on this particular beta carbolene studies. And we did various, as I mentioned, conjugation with, again, benzimidazoles, dithiocarbanates, and so on and so forth. And you can see that a large number of these molecules showed wonderful activity with respect to the topoisomerase 1 inhibition also. And we have also some of the, conjugate I mentioned with the DNA intercalating agents, which also helped us in coming out with some very new molecules. So in this whole effort, I think I'll just skip this and come back to, yeah, this is again beta carbolene with cumbrastatin. So you can see we tried to bring in two scaffolds together also. When we talked of, these are the two natural products again. And interestingly, good cytotoxicity. It induces apoptosis. And then this is, you can see once you bring in two together, it's becoming two topoisomerase 2 inhibitor. And quite a lot of studies were done on such molecules, where in variations of this, we could get topoisomerase 1 also in some of these, once the cinamido type of linkages were brought in. I think I can. With simple molecules like chalcones also, we could bring in together. And these are the beta carbolene hydrantoin hybrids. And these are benzimidazole conjugates. These are the imidazopyridine analogs. So in summary, I could say that although today making conjugates and hybrids have become a very important technique. And initially, when we started this, we didn't realize. That was the time when not people were looking into it. So this is the thing which we started almost 20, 25 years back, making such type of hybrids. And lots of drugs have now come in the market with such a strategy. That much I can tell you. And these are only few names. There must be much more who contributed in this whole exercise. This is 2016 when they assembled for a conference which was done in my honor at IICT. So a large number of my past students, present students, and so on had come from various places to attend this meeting. So practically they had a group photograph in front of my laboratory building only. So uh, this is the laboratory. Maybe some of you must have seen the Discovery Building in ISCT, and this is where on the first floor I had my laboratory. I mean, and in fact, on the other side, I went in for maybe in 2012, 13 for another floor because the labs were not sufficient for taking up some of the uh, not only biological but also many other detailed studies. So we went for another floor on that, in fact, later. So presently I'm with BITS now. Mostly I am attached to pharmacy, but it's mostly mentoring the faculty of pharmacy, chemical sciences, and biological sciences. So it's a wonderful institution, I would say. So thank you all for your listening, and I'll be happy if you have some doubts, some questions.
While attaching the side chain, like 3, 4 carbons, is there any chances of isomers are obtained?
No, see,
So what?
I didn't go into the synthesis of that for various reasons. Otherwise, it would take hours together. The attaching has to be done before you go to the critical steps. So when you're using your starting subunits, you do the attachment before. Otherwise, if you do the attachment later, you will not get your final product at all. You may go into so many side reactions.
Yeah.
Your question is perfectly correct. So we never go there, do that way. So particularly when you have an imine. Imine is so reactive, you can't handle it after it's formed. You can't do any other reaction on that later because it reacts immediately, and you want that imine to be in place. So before building that, you start with your subunit, starting material, attach them with all these open precursors, and then do the next sequential steps. That's how it goes. Otherwise, you never think of doing such a
Yeah.
right.
And also you attach biphenyls, right? So for only how you attach only for particular fourth and fourth position. So for the four
You start with that. You start with that. First, do that, and then finally link it at that hydroxy linkage or an amino linkage. You can't do later. It's always to be done before. Most of these precursors also, the subunits of conjugates what we have prepared, you prepare them first to certain, or you don't close. I mean, suppose there are sensitive groups, you don't do that. So you do in such a way. Together you do it, or you do step by step in such a way without harming the other side groups. Then only it's practically possible. Otherwise, it's not.
And also the molecular weight of all these natural persons.
Pyrrolobenzodiazepines, this is an exception where these are in nature itself found. These are large molecules. But once you do dimers, you are getting an advantage of it's becoming much more bulkier, but from 3 base pairs, you are spanning to 6 base pairs. So the selectivity, I didn't go into those details. Selectivity has definitely helped to be selective. As I said, not sequencing any of the compounds from between because you don't have a clarity, 100% clarity. Although the human genome project was done in late 80s and early 90s, that has helped a lot. Way one can do. We are not able to get that type of a clear differentiation. So what we generally do is when we carry out the cell line assays with respect to cytotoxicity, you also use a normal cell so that you know that whether this particular molecule is it less toxic to normal cell line or not. If it is too toxic, you don't proceed with that. It's of no value because it's as good as killing somebody. If a cancer patient may be better off without taking a drug of that type because otherwise it's like a total poison. So that is why you do have to balance this. It should have some safety level for normal cells.
Such a big molecule, active site pocket may not hit this big molecule. So only you will get some side chain interactions.
Achha,
The total molecule.
did you see my previous one?
Yeah, I saw
That
it.
double one?
Yeah.
The double one gets inside the minor groove completely in the end. Because of the shape of that molecule. It blends along with the groove.
So maybe the depth of that groove helps to accommodate this.
I can go, it's not there. When this dimer of pyridobenzene is being posed, that's how it is having 6 base pair sequence selectivity and cross-linking at 2 strands of DNA. If it doesn't go in, you cannot get that crossing. Because a lot of X-ray studies were also done along with this formation to prove this, and also some NMR studies, NOSI studies, and all that to understand how they get inside. A lot of studies have been done, not only by us, but many another 2-3 groups have been actively working on that in those days. From one of the group from UK, another one from US, another group from another European country. So this has that molecule now. What I said that SJT from the UK side, they have been FDA has approved that for antibody they're using it as a toxic load for antibody drug conjugate, and it is being used today clinically, that molecule. So it's a huge molecule, but it's such a targeted one they are doing through antibody drug conjugate. It is being clinically used. I think just 1.5 years back, the FDA had approved it. Yeah, please.
Actually, the opposite what we have discussed earlier.
Pardon, sorry, I don't.
Opposite what we
Sure.
have discussed.
Hmm.
Right. It has less solubility as well as less permeability. So for that possible.
No. Solubility is not a problem because photoprotoxin is not soluble.
Okay, but
Etoposide,
bioavailability.
once you have this, it is soluble. That is the purpose it has been made. It is soluble. But there are other molecules other than photoprotoxin which are much more active. But solubility, what they have done is, even we have done it, at the pendant ring on the OH, you make a phosphate salt to make it soluble. Many of these natural products, modified ones, many are trying as phosphate salts, and some have been successful. So, bioavailability as well as solubility improves. Yeah, maybe you were asking something more.
My question was, being from Sudik's background, I just wanted to know how to come up with the novel drug delivery system and what could be the challenges that a formulator could face while he is coming up with such kind of drugs.
As I told you, in case of cancer, the most important, we have all, I did come out, show some of the results of our proton monotherapy once, which is again a selective one, where we have led this molecule as a conjugate in such a way that once it releases, it becomes immune. And it goes to the site. There, with that specific enzyme, it is more of hepatocellular type of cancer, because that is more predominant in a living system. So some animal studies were also done. So such things are possible. Where, and as I said, the most important thing which is happening is, but it is very, very expensive and the expertise is not there everywhere for the antibody drug. And another important thing which is coming to the today is a lot of people are working on programs. So this is again a very important area which is coming, and many companies, many industries are racing for this. Both antibody drug conjugates, I think today 8 or 10 molecules of antibody drug conjugates have been approved by till last year from the point of view of clinical usage, and close to 200 ADCs. So you can imagine that quite a lot of things will come in coming years where the therapy is going to be much safer, selective. And as I also mentioned, there is another concept which is being used, particularly in Western Europe, where they are doing along with immunotherapy, so that the patient immune system doesn't go down. So it's a combination of both anti-cancer agents, immunotherapeutic agents, and the immunotherapy together, where you keep the patient with lesser side effects. So these are some things which are very useful. As you said, a very simple technique to make the program is one thing. But these things, the advanced things, I'm sure it's not going to be done in each and every level. Because even certain big companies also do it with a lot of, I would say, so it's a big investment also. Because anywhere, wherever a lot of biologics and immunoglobulins, antibodies are involved, the cost to itself is very high. So the project also is very expensive. There are various, because today a lot of in silico uh, so all these things are possible.